This is Lecture 5, Neuronal Adaptation and Communication, and it corresponds with Chapter 5 of your textbook. Okay, getting into the course material, this corresponds to Chapter 5, Section 1, Chemical Messages. So we have two main types of chemical messengers that we're going to discuss, and those are neurotransmitters and hormones. Neurotransmitters are chemicals that signal in the central nervous system and have an excitatory or an inhibitory effect on the target when they're released on a neuron. There are maybe 60 to 100 different neurotransmitters in the central nervous system. It can be kind of difficult to decide what's a neurotransmitter, so scientists are still trying to work out exactly what's a neurotransmitter and what isn't. Then hormones are chemicals that are transported by the circulatory system, and they act on organs, but they also affect behavior and general body physiology. The hypothalamus controls pituitary function. It controls the pituitary gland. The pituitary gland releases hormones into the bloodstream, so the hypothalamus is kind of the one ultimately controlling hormonal release. Then hormones affect target glands and organs in the autonomic and enteric nervous systems. Hormones do travel a lot further than neurotransmitters, but they're also slower acting.
These are just some major neurotransmitters in the body, and we're going to be returning to the specific roles of these, but you've probably heard of things like dopamine, maybe glutamate, norepinephrine, which is noradrenaline, serotonin, things like this. So getting back to the neuron, if you remember, the synapse is this junction between two neurons, between the presynaptic neuron, which is the neuron that's releasing neurotransmitters, and the postsynaptic neuron. A chemical synapse is this junction where messenger molecules or neurotransmitters are released from one neuron and they excite or inhibit the next neuron. Most synapses in the mammalian nervous system are chemical, and when we talk about synapses in general in the human nervous system, we're almost always talking about chemical synapses. So, as mentioned before, the presynaptic membrane, which is also the axonal terminal, is where the action potential terminates to release the chemical message. And the postsynaptic membrane is after the synapse. This is also typically a dendritic spine, although it can be other parts of the neuron, is the receiving side of the chemical message. So this is where neurotransmitters get released and excitatory postsynaptic potentials or inhibitory postsynaptic potentials are generated. The synaptic cleft is this space between the two neurons. So there's just a very small amount of space. These two neurons are not actually touching, and so the small gap is called the synaptic cleft. Synaptic vesicles are presynaptic. They're small membrane-bound spheres that contain one or more neurotransmitters. Storage granules are also presynaptic, and they're membranous compartments that hold several vesicles. And then postsynaptic receptors are these sites, these proteins that are embedded in the postsynaptic membrane, and these are the sites to which a neurotransmitter molecule binds. And these are just some pictures of synapses from electron microscopes. So these little things that look like bubbles are vesicles, and these contain neurotransmitters in the presynaptic cell. And these gaps you see between the cells, that's the synaptic cleft. So there is a different type of synapse that is much less common than chemical synapses in mammalian systems, and that's electrical synapses or gap junctions. These are ion channels that form a pore through which ions can flow directly, so there's no cleft or no gap between the cells. This transmits information faster than chemical synapses. It is one way that interneurons fire in synchrony, so fire together by using these really quick electrical synapses. It allows for exchange for substances between neurons and glial cells, but chemical synapses are more common in the mammalian brain because they're more flexible, complex, and they can change with experience. So learning and memory are dependent on chemical synapses. So within a kind of chemical synapse, there are 4 steps for neurotransmission, or the transmission of neurotransmitters into the synaptic cleft for reception on the postsynaptic cell. The first step is synthesis and storage. So some neurotransmitters are synthesized in the cell body and transported to the axonal terminal, and others are synthesized right in the axon terminal from raw materials that came from the cell body. The second step is the release of neurotransmitters. When an action potential reaches the axon terminal, it triggers voltage-sensitive calcium channels to open. Calcium ions flow into the presynaptic terminal, and this influx of calcium ions causes the vesicles to fuse to the presynaptic membrane. And the vesicles, once they fuse with the membrane, they kind of push out their contents into the synaptic cleft, so now there are neurotransmitters in the synaptic cleft. And this is just an image from an electron microscope. You can see where the little arrows are pointing that vesicles have fused with the cell membrane and are pushing their neurotransmitters into this gap between the cells, the synaptic cleft. In the third step of neurotransmission, the postsynaptic receptor site is activated, so the neurotransmitters diffuse through this synaptic cleft, this gap, and bind to the receptors on the postsynaptic cell. Receptors are specialized protein molecules that are embedded in this membrane on this postsynaptic cell. And the postsynaptic cell responds in one of 3 ways. There can be an excitatory response, there can be an inhibitory response, and there can also be kind of a setting off another chain of chemical responses that are ultimately excitatory or inhibitory. So if you remember, in the nervous system, there are kind of like 2 main types of responses or ways of transmitting signals, and that's excitatory and inhibitory. So ultimately, all actions between neurons are excitatory or inhibitory. Okay, and this is just another image of the activation of receptor sites. The neurotransmitter is released into the synaptic cleft. It binds to the receptor. It's kind of like a key in a keyhole. The neurotransmitter doesn't actually go into this postsynaptic cell. It just binds to this receptor, which creates a response. So the 4th step of neurotransmission is that the neurotransmitters are deactivated. So after the neurotransmitter has bound to its receptor, one of 4 things can happen. Either the neurotransmitter diffuses away from the synaptic cleft, it just drifts away. It can degrade in the synaptic cleft, so enzymes or other chemicals can break down that neurotransmitter and make it inactive. Another common one is free uptake, so the neurotransmitter is transported back into the presynaptic cell for reuse later. And then astrocytes, which if you remember are glial cells, take the neurotransmitter and typically store it for later use. There are a number of types of synapses. For simplicity, I don't expect you to know all of these. For the purpose of this course, we're mostly going to be talking about axodendritic and axosomatic synapses. So axodendritic are the ones where an axon synapses with a dendritic spine or any part of the dendrite of another cell, and axosomatic is when an axon of one cell synapses with the soma or the cell body of another cell. Excitatory synapses are typically axodendritic, so typically when an axon is synapsing with a dendrite, that's more likely to be excitatory, whereas inhibitory synapses are typically on the cell body.